Hello to Prof. Dr. Wan Kelan. This is a seminar video presentation from Group 6. As you can see on the screen, article that we choose is a chromosome-level genome assembly of Hong Kong catfish, Clarias fuscus, uncover a sex-determining region.
Group 6 members, Eugene, Diana, Shahirah and Rafika.
Let's move to introduction part. Hong Kong catfish, or the scientific name is Clarias fuscus, is an ecologically and economically important species that is widely distributed in freshwater region of southern China. Hong Kong catfish had significant sexual growth dimorphism. Sexual growth dimorphism is the differences in appearance between males and females of the same species, such as in color, shape, size, and structure. Under the same breeding condition, the growth rate of Hong Kong catfish is significantly higher in males than in females. Clarias is an excellent model for understanding the evolution of sex determination because the gene includes species with an unusual diversity of sex chromosome system. The objective of this research is to determine the chromosomal-level genome assembly of Hong Kong catfish. The monosex fish culture of Hong Kong catfish has important economic significance. The lack of genomic research for this species is serious impediment to monosex breeding. Thus, it is necessary to obtain the chromosome-level genome of the Hong Kong catfish as a resource for molecular breeding. The first method we need to do is preparation of the sample. Sample is obtained from the Guangxi Introduction and Breeding Center of Aquaculture, 1 male and 1 female with similar sizes. Sample is with anesthesia in a bath of ethanol. Dissection is performed and stored at 4 degrees C in boiling fixative solution. All parts of the sample were collected, snapped, frozen in liquid nitrogen, and stored at negative 80 degrees Celsius. Then, genomic DNA is extracted from muscle tissue of male fish using Stbc method. The concentration and quality is examined using nanodrop 2000 spectrophotometer and 0.8% agar gel electrophoresis. Stbc method is commonly used method to extract DNA tissue use the ionic detergent cetyl trimethyl ammonium bromide. Next step is use of triazole method to identify the total of RNA was extracted from male and female fish. This is the general method for deproteinization RNA. Last one is RNA integrity and purity were examined using nanodrop 2000 spectrophotometer and bioanalyzer 2100 system, and to extend RNA degradation can examine using 1.5% of agar gel electrophoresis. Next method is we need to do library construction and sequencing. First, genomic backfire sequencing library were constructed using the SMRT Bell Express Template Prep Kit 2.0. This sequencing libraries is to determine the concentration of fragment size distribution of sample using the femto pulse system and qubit 3.0 fluorometer. Next is fragment size selection is performed using blue pimpin system to remove sequence fragment below 25 kilobase. Then for construction of Hi-C library, 1 gram of muscle tissue is used to prepare the library. The Hi-C library was sequenced on the MGI Sequence 2000 platform. Small fragment genomic library is constructed using Vault's Universal DNA Library Prep Kit. The concentration and fragment size distribution is determined by qubit 3.0 fluorometer and bioanalyzer 2100 system. After the library is being tested, sequencing was performed on the MGI Sequence 2000 platform. HTQC is used to remove adapter, duplicate reads, and containing sequence and low quality reads. Lastly is libraries of whole tissue mRNA from males and females is constructed using Vault's Universal V6 RNA sequence library. Kit library quality is determined using qubit 3.0 fluorometer and bioanalyzer 2100 system.
For the first method is genome assembly. A preliminary genome assembly obtained using MiCap for pack biosequencing. After the assembly was completed, the error correction was performed in order to improve the accuracy of assembled genome by identifying and correcting the error. Next, small fragment data were used for genome polishing using Pillen. Finally, the content-level genome assembly was obtained. The genome Hi-C sequencing data were compared using BWA-MEM and a single end reads and sequence beyond 500 base pair from the enzyme cut site were removed. Next, filter Hi-C data were used for chromosome construction. Hi-C is a technique that captures the spatial proximity of DNA fragment within the nucleus. It can be used to identify physical interaction between DNA segments on the same chromosome. By integrating Hi-C data with genome assembly, it is possible to scaffold contigs into larger fragments correspond to the catfish chromosome. So proximity information provided by Hi-C helps order and orient contigs, creating the chromosome-level scaffold. Next, the contigs were clustered, sorted, and orient using 3D DNA to obtain the chromosome-level genome, as I said. Next, the chromosome-level genome was visualized and error-corrected using JuiceBox. And finally, the completeness of Hong Kong catfish genome assembly was assessed using BASCO based on the single-copy homologous gene set. For the next method is genome prediction and annotation. The homology base and de novo annotation were used to identify repetitive sequence in the genome. So for firstly, transposable element sequence were searched from the rep database based on the homology using repeat masker and repeat protein mask. Repeat modeler and LTR finder were used to construct the Hong Kong catfish repeat sequence database de novo and then identify to constructed database using repeat masker. TRF were used to identify tandem repeat sequence. The result were integrated and the final annotation result of duplicate sequence were obtained by removing the non-redundant part after overlapping. Alright, as we know that the non-redundant means the region of the genome that contain repetitive sequence or duplication. By removing the non-redundant part, the gene prediction algorithm can actually focus on unique region of the genome, improve the accuracy of the gene predictions, and reduce the false positive and false negative annotation. Okay. Next, the homology, de novo, and transcriptome annotation was used to predict the structure and function of protein coding genes. Okay. So, the coding gene annotation information of all of these species was selected for homolog-based annotation. Augustus and GenScan was used to de novo annotation. Top hat was used to match the transcriptome data to the Hong Kong catfish genome. The maker was used to integrate the gene set obtained by various methods into a non-redundant gene set. And then the annotation information for non-coding region and variable shares were added using PASA. The protein in the gene set were functionally annotated with protein database such as the SwissProt, the Tremble, CAG, Interpro, Go database, and NR. So the tRNA scan was used to find the tRNA sequence in the genome. Infernal in RFAM was used to predict miRNA as an RNA sequence in the genome.
Next method is genome evaluation analysis. Gene family clustering was performed based on protein sequence of Hong Kong catfish and other 15 fish species. By using software Ortho MCL, the protein sequence for each species were clustered based on sequence similarity. The phylogenetic tree were constructed and it based on the shared single copy direct homologous gene that obtained from gene family clustering. Using RAX-ML, the phylogenetic tree were constructed by the maximum likelihood method. The divergence time was predicted using MCMC3 and time tree for calibration. Next, random birth and death processes were to stimulate gene family expansion and contraction for each lineage on the phylogenetic tree, and this step used software called catfish. Chromosomal genome included in colorimetry analysis by using Murmur. Next, identification of the sex determination region and potential sex-related gene. A high-density lineage map of Hong Kong catfish were constructed with SNP marker and the sex-linked QTL was identified with a total map distance. SNP markers from the genetic lineage map were mapped to the chromosome-level genome using BWA software. This step is to determine the correspondence between the lineage group of the genetic lineage map and the chromosomes of the genome assembly. The genomic region corresponding to the sex-linked QTL obtained based on comparison of the 2 to 5 SNP markers. Then, the number of the gene in the sex-linked QTL and the annotation information obtained and gene function was obtained by SwissProt, PubMed, and CBI databases.
For the first result is genome sequencing and assembly. The PacBio data were used to construct the preliminary genome assembly. The size of preliminary genome assembly was 966.34 megabase, including the 897 contigs, and the contigs N50 were 8.18 megabase. The interrupted contig were sought to obtain the chromosomal level genes. The Hong Kong catfish chromosome level genome assembly was obtained with a length of 933.4 megabase, and the 416 contigs, which equal to 28 chromosome, and the contig N50 were 8.52 megabase, and the scaffold N50 were 35.68 megabase. Alright. So, according to the BASCO result, the genome contained 2,493, including the 2,405 single copy and 88 duplicate. So, the result indicate that the genome assembly has high coverage and the completeness. Next is genome annotation. So table 2 shows that the statistical summary of protein coding gene prediction in the Clarias fuscus genome. So in the column software, the gene number of PASA software was 23,345, and the average gene length is 19,000, more than 19,000. So out of this gene number, 18,493 gene encoded single transcript, and the 4,852 gene encoded multiple transcript. So the characteristic of functional gene were compared with those of other catadromous fishes. So the gene characteristic of Hong Kong catfish actually similar to the siluriforms. So the gene were annotated by the protein database, as I mentioned before. Under the DNA transposon were most common TE type in the Hong Kong catfish genomic, followed by the long terminal repeat and the long interspersed nuclear element. So the tRNA scan, BLAST-N, and rFAM used to predict non-coding RNA, and the total of tRNA was 12,110, RNA 1,672, snRNA 506, mRNA 278 were predicted in the Hong Kong catfish genome.
Next is the result of genomic evolution analysis. Gene clustering was performed based on protein sequence of Hong Kong catfish. 15,977 gene families and 3,183 single copies genes were identified. Gene families of 16 fishes were compared. A total of 60 unique, 353 expanded, and 851 contracted gene families were identified. By phylogenetic analysis, it can be concluded that the Hong Kong catfish and sea maggot clustered in a single branch. Hong Kong catfish closely related to fishes belong to other siluriformes. Hong Kong catfish diverged from sea maggot about 63.7 million years ago. All 28 chromosomes of Hong Kong catfish smaller than 29 chromosomes of Ipentatus and 30 chromosomes of P. hypothalamus. Hong Kong catfish chromosome 1, 9, 12, and 14 is underwent chromosome breakage and recombination that found in chromosome 3 and chromosome 5. Next is the result of identification of sex determination region. From table 1 shown, the result shows that each lineage group of genetic lineage map corresponds to one chromosome of the genome assembly. And among 785 genes in the sex-linked QTL, 18 sex-related genes were identified. It can be looked at table 2. Some genes identified including Fanconi anemia, group M, or the gene is a FANCM, and domain containing protein 2 and others. The sex-linked QTL was located on chromosome 13 of the genome assembly. Based on the gene annotation result for chromosome 13, 785 genes were identified in sex-linked QTL. The graph shows the location of the sex-related genes in the QTL intervals of Clarias fuscus. The lower abscissa in genetic distance in centimorgan. The upper is the genomic position in megabases. The ordinate is the LOD threshold. Black dots are SNP markers. Brown dots are sex-related genes.
From the results of chromosome-level genome assembly, the scaffold N50 value of C. fuscus is 35 megabase, which is higher compared to other siluriformes catfish, which indicates the constructions of this Hong Kong catfish genome assembly is of high quality. The sex-linked QTL is found in the chromosome 13, thus it is speculated that chromosome 13 is the sex chromosome of Hong Kong catfish. The genome comparison results show that some sex-determining genes in other siluriformes fishes were not mapped to chromosome 13 of Hong Kong catfish. This may be due to the fact that the genomes of siluriformes fishes are generally large and multiple genome duplication events have occurred. Based on the gene annotation results for chromosome 13, there are 18 sex-related genes were identified in the sex-linked QTL, and among them, 5 infertility-related genes and 13 sex-related genes associated with gametogenesis were identified. These genes were detected in other species such as humans and mice, and they show abnormalities in fertility and gametogenesis when these genes are lagged, knocked out, or overexpressed. The chromosome-level genome and sex-linked loci is useful for the development of sex-linked markers that can be used to identify male juvenile catfish, then we sex-reverse it, bred with normal male to produce single male Hong Kong catfish family groups. The sex-linked loci also allows identification of sex-determining genes that can be knocked out to produce single-sex Hong Kong catfish population. So in summary, the construction of chromosome-level genome and identification of sex-linked loci can promote the development of the monosex culture industry of the Hong Kong catfish that has important economic significance.
From this research article, what we can conclude is the chromosomal-level genome assembly of Hong Kong catfish was determined. The continuity and completeness of the Hong Kong catfish genome were consistent with those of other high-quality teleos fish genome. A sex-linked QTL was focused on chromosome 13, which was predicted as the Y chromosome, and 18 genes in this region were considered as sex-related genes. That's all from us, Group 6. Thank you.